Hi everybody, welcome to the video lecture on blood vessels. In this video we will outline the anatomy of the major blood vessels and later move on to the physiology. We have covered briefly in the cardiovascular system what is the difference between arteries versus veins. We know that arteries are going to be vessels that conduct blood away from the heart, and veins by definition are going to be vessels that are going to take blood back to the heart. What you see depicted here is the entire system of vessels incorporating the heart. So we've already talked about how blood moves within the heart. Now we want to look at a more holistic approach where we see that when the ventricles, the left ventricle contracts, it sends the blood to the aorta and to the major conducting vessels. So we know that the aorta branches, and every time that major vessel splits, it becomes a different type of artery, as you will see in a future lab. So from the aorta, we have elastic arteries. So the aorta is actually a type of elastic artery, sometimes known as conducting arteries. Well, these arteries then become smaller, if you will. Their diameter gets smaller the further away you get from the heart, and eventually these arteries are going to become smaller, known as arterioles, and then finally capillaries. By definition, a capillary is an exchange vessel. It's where nutrients are going to be exchanged, and you also have removal of waste products such as carbon dioxide. From the capillaries, the next thing that happens is that these capillaries are going to remove all the waste products from the tissues or the organs, meaning diffusion happens from the tissues into the capillaries, and then the blood enters what we call the venous pathway, where you have the smallest veins known as venules, and then of course we've learned that the largest vein is going to be the vena cava. The other thing that you see in this image is the incorporation of the lymphatic vessels, which we will cover in a future chapter. So at least this gives you a good overview of exactly how the pathway of the blood moves as you go through the entire body. And again, we will name selected vessels, arteries, and veins when we cover these in lab. So let's move over now and look at the difference in the tissues that make up the major arteries and veins and also capillaries. Let's begin with arteries and veins first, and then we will end with capillaries. Both arteries and veins are made up of three tissues known as tunics. So we name them according to whether they're in the innermost or if they're lining the external. So we'll start with the innermost, known as the tunica intima. The tunica intima is made up of endothelium, and when you hear the word endothelium, think simple squamous epithelium. So because this is lining it's found in the innermost portion of the vessel, we know this as endothelium. The lumen is going to be the space whereby blood travels. So both arteries and veins have lumens. Note one difference. So that we're going to look at some differences between the two vessels. So the first difference that we know is that the lumen on an artery is going to be smaller than the lumen on the vein. And recall that the structure is always going to fit the function. There's a reason why the lumen in a vein is a lot wider, meaning the space, versus that of an artery. So besides having the endothelial cells, some of these vessels also contain a subendothelial layer, which is typically found in vessels larger than 1 millimeter in diameter. The subendothelial layer consists of a basement membrane and loose connective tissue, which is going to support the endothelium. Let's go over to the next layer, which is the middle layer, known as the tunica media. The tunica media is primarily composed of smooth muscle and also elastic fibers. It's important to note your second distinction is that if you look at this image, the smooth muscle layer in the artery is going to be a lot wider than the smooth muscle layer in a vein. And this is because arteries are associated with higher blood pressure than veins are. So it's important that the vessels, the arteries, can regulate the widening and the closing of the lumen. We know this as vasoconstriction and vasodilation. So vasoconstriction happens when the lumen gets a lot smaller or narrow, And vasodilation, as we've learned already, happens when the vessel is going to become wider. So that's the primary reason why you have a larger smooth muscle layer in an artery than you do in a vein. The last layer is known as the tunica externa, which is primarily composed of collagen fibers. These collagen fibers are going to protect and reinforce the vessel and anchor it to surrounding structures. Now, the tunica externa is also infiltrated with nerve fibers, lymphatic vessels, and in larger veins, a network of elastic fibers. In large vessels, the tunica externa contains a system of tiny blood vessels known as the vasa vasorum, which literally means vessels of the vessels, and these nourish the more external tissues of the blood vessel wall. The innermost portion of the vessel also obtains nutrients directly from blood in the lumen. Now, the last type of vessel is going to be the capillary. So the capillary, as we outlined earlier, is the exchange vessel where diffusion of nutrients, gases takes place. Note that the capillary is only made up of one layer of endothelial cells, and it's also outlined by the basement membrane. So again, this makes sense because if the capillary has to deliver, or it's the site for exchange of nutrients and waste products, we want that layer relatively thin because that is going to facilitate the tissue exchange or the diffusion of the nutrients and so on. In the first slide of this PowerPoint, you see a histology slide comparing the arteries versus veins. Now you see the two differences. Histologically, we see a wider lumen in veins versus arteries, and then we see a wider band of smooth muscle in the arteries versus a vein. And again, we will outline that more specifically as we unravel this lecture. So let's talk now about the variation in the arteries. So let's begin with elastic arteries, which are thick-walled arteries near the heart. The aorta and its major branches are included. These arteries are the largest in diameter. They can range from 2.5 centimeters to about 1 centimeter. Elastic arteries are oftentimes referred to as conducting arteries as well. You can think of these arteries as pressure reservoirs because they expand and they recoil as the heart ejects the blood. Now, the blood vessels here are meant to move blood in a continuous manner rather than starting and stopping with the pulsating rhythm of the heartbeat. You should read in your chapter of the homeostatic imbalance, which would be atherosclerosis. So atherosclerosis is a kind of arteriosclerosis. So arteriosclerosis is one of many proliferative and degenerative changes in the arteries, leading to their decreased elasticity, whereas atherosclerosis is the most common form of arteriosclerosis, where small patchy thickenings called atheromas form that can intrude in the vessel lumen, making it easy for arterial spasms or a blood clot to close the vessel completely. So of course, because of the variation in our diet and nutrition, atherosclerosis is something that's very important to go back to and look at. You can think of the vessel kind of like a clogged pipe. So when we have a draining system in our bathroom and maybe it's clogged by hair or other debris, of course the water is going to not be able to drain with ease. And it's the same thing that can happen in atherosclerosis, where you can have the clogged artery is accumulating all the lipids that are consumed in the diet, hence it's going to obstruct that vessel. So some of the risk factors, of course, are going to be age. As we age, we have more of a predisposition. Being male, family history, high blood cholesterol, also hypertension, smoking cigarettes, lack of exercise, diabetes, obesity, stress, and having a high amount of trans fats in the nutrition. So it's important to go through some of the preventative options and even treatment options. So preventative measures would be avoid smoking, trying to lose weight, exercise regularly so that you can increase blood levels, and also increase the high density lipoproteins such as HDL, also known as the good lipoproteins, and of course having a healthy diet which is low in saturated and also trans fats. Let's move over to the muscular arteries next, which are going to deliver blood to specific body organs. Sometimes we refer to these muscular arteries as distributing arteries because they account for most of the named arteries studied in our anatomy lab. Their internal diameter ranges from that of the little finger to about the width of a pencil lead. So proportionally, muscular arteries have the thickest tunica media of all vessels. Their tunica media contains relatively more smooth muscle and less elastic tissue than do the elastic arteries. So for this reason, they are more active in vasoconstriction and less capable of stretching. In muscular arteries, however, there is an elastic membrane on each face of the tunica media. Let's go over now to the arterioles. So the arterioles are the smallest of all the arteries. They have a lumen diameter ranging from about 0.3 millimeters down to 10 micrometers. The smaller arterioles, which lead into the capillary beds, are little more than a single layer of smooth muscle cells around the endothelial lining. Now, the flow that blood is going to take into the capillary beds is determined by arterial diameter, which varies in response to changing neural, hormonal, and local chemical influences. Changing diameter changes resistance to blood flow, and so arterioles are often called resistance vessels. So when arterioles constrict, the tissues served are largely bypassed, and when arterioles dilate, the blood flow into the local capillaries increases dramatically, and we'll take a look at this in a future slide. So this figure summarizes the major arteries that we just talked about, elastic arteries with their diameter and lumen. You also have the muscular arteries and the arterioles, along with the relative tissue makeup. Now what we're going to do is we're going to move over to capillaries, which we said are going to be these microscopic exchange vessels. They're actually the smallest blood vessels that we have in the body. They have thin walls that is just the endothelium, which is the tunica intima, as we earlier explained. In some cases, or most cases, one endothelial cell forms the entire circumference of the capillary wall. At various locations along the outer surface of some capillaries, there are these spider-shaped cells known as pericytes, which are contractile stem cells that can generate new vessels or scar tissue, and thereby stabilizing the capillary wall and help control the capillary permeability. The lumen diameter of a capillary is typically between 8 to 10 micrometers, which is just large enough for red blood cells to pass through. And most of your body, most tissues have rich capillary supplies, but there's few exceptions. You probably recall this from the skeletal system, anatomy and physiology, that tendons and ligaments are poorly vascularized, and that's the reason why they heal so poorly. Cartilage and epithelial lack capillaries but receive nutrients from blood vessels in the nearby connective tissue. There are three types of capillaries that we need to review. These are the continuous capillaries, the fenestrated, and the sinusoids. So the first thing that we need to note is that all three types of capillaries have tight junctions that join their endothelial cells together. However, the junctions are usually incomplete, and they leave gaps of unjoined membrane called intercellular clefts, which allow limited passage of fluids and small solutes. And there are some capillaries that are more leaky, which have specialized passageways that increase fluid movement. So let's begin with the first one, which are the continuous capillaries. These are the least permeable and the most commonly found. They're abundant in the skin, the muscles, the lungs, and also the central nervous system. They often have associated pericytes, which are those contractile stem cells that can generate new vessels, and they can stabilize the capillary wall and help control the permeability. Let's move over to the second, which are the fenestrated capillaries. These have large fenestration. So a fenestration is a pore, and thereby the pores enable for increased permeability. These occur in the areas of active filtration, such as in the kidneys, or anytime you have absorption, like in the small intestines, and areas of endocrine hormone secretion as well. They kind of remind me of Swiss cheese because they have these holes that tunnel through the endothelial cells as well. Let's move over to the next type, which is going to be the most permeable, and these are the sinusoid capillaries. They are occurring in very limited locations, such as in the liver, the bone marrow, spleen, and adrenal medulla. You may remember this from the blood chapter or video where we talked about the sinusoids, and when white blood cells are originated in the bone marrow, they're able to leave that site via these sinusoids. So that's what we were talking about. These capillaries also have incomplete basement membranes. They are irregularly shaped and have large lumens in comparison to the other two types of capillaries that we discussed. Now, capillaries do not function independently. Instead, they form interweaving networks called capillary beds. The flow of blood from an arteriole to a venule through a capillary bed is called the microcirculation. In most body regions, a capillary bed consists of two types of vessels. You have a vascular shunt, which is also known as a medarterial thoroughfare channel, which is a short vessel that directly connects the arteriole and venule at opposite ends of the bed. You also have the second type, which are true capillaries, and these are the ones that we know of as the actual exchange vessels. So if we look at this image, we could see the terminal arteriole feeding the bed leads into meta-arteriole, which is continuous with the thoroughfare channel. The thoroughfare channel in turn joins the postcapillary venule that drains the bed. And the true capillaries number about 10 to 100 per capillary bed, depending on the organ or tissue served. They usually branch off the medarteriole, proximal end of the shunt, and return to the thoroughfare channel, which is on the distal end of the shunt. But occasionally they spring from the terminal arteriole and empty directly into the venule. A cut of smooth muscle fibers called the precapillary sphincter surrounds the root of each true capillary at the medarteriole and acts as a valve to regulate blood flow into the capillary. And blood flowing through a terminal arteriole may go either through the true capillaries or through the shunt. When the precapillary sphincters are relaxed, meaning that they're open, blood flows through the true capillaries and takes part in exchanges with tissues. When the sphincters are closed or contracted, blood flows through the shunts and bypasses the tissues and cells. So to take another example, when you exercise vigorously, blood is rerouted from the digestive organs to the capillary beds of your skeletal muscles where it is more immediately needed. So this rerouting helps explain why vigorous exercise right after a meal can cause indigestion and also those famous abdominal cramps. So now that we've outlined the arteries and the capillaries, let's move over to veins, which we said are going to be the vessels that are going to take the blood back towards the heart. So capillaries unite to form venules, which are smaller veins, and they range between 8 to 100 micrometers in diameter. The smallest venules are known as postcapillary venules. They consist entirely of endothelium, around which pericytes congregate. Postcapillary venules are extremely porous, more like capillaries than veins, and fluid and white blood cells move easily from the bloodstream through their walls. And larger venules have 1 or 2 layers of smooth muscle cells, and a thin tunica externa as well. And as we depicted or illustrated earlier, venules, of course, are going to join to form veins. Veins usually have all three distinct tunics that we talked about, but their walls are always thinner and their lumens, of course, are a lot larger. So the reason why veins have larger lumens is because veins are called capacitance vessels or blood reservoirs because they can hold up to 65% of the body's blood supply at any time. And looking at this image, you could see the relative proportion of blood volume through the cardiovascular system with the systemic veins and venules accounting for that 65% of the blood supply. So another feature of veins that are not present in the arteries are going to be valves, the venous valves, which prevent blood from flowing backward in veins, just as valves do in the heart. And they represent another adaptation to compensate for low venous pressure. So these valves are formed from folds in the tunica intima, and they resemble the semilunar valves of the heart as mentioned earlier. So here's just an image going back to what we talked about between arteries and veins. You could see the valves, and this is what we are referring to. So venous valves are most abundant in veins of the limbs where gravity opposes the upward flow of blood. They are usually absent in veins of the thoracic and abdominal cavities. If we look at a homeostatic imbalance, some of you know what varicose veins are, which are really going to be these veins that are dilated because of leaky valves. So about 15% of adults suffer from varicose veins, usually in the lower extremities. Several factors can contribute, such as heredity and conditions that hinder venous return, such as when you stand for a prolonged period of time. It could also be due to obesity and pregnancy as well. So consequently, what happens is that blood pools in the lower limbs, and with time the valves weaken and the veins, the vein wall stretches. So superficial veins, which receive little support from surrounding tissues, are specifically susceptible. So just like we highlighted in or reviewed by looking at an image of the main arteries, here is a summary of the veins, and we talked about the average lumen, diameter, the relative tissue makeup as well. Let's move over to arterial or vascular. Let's start with vascular anastomosis, which means that blood vessels are forming these special interconnections. So since most organs receive blood from more than one arterial branch and arteries supplying the same territory, often merged together, they are going to form the arterial anastomoses, which provide alternative pathways known as collateral channels for blood to reach a given body region. So if one branch is cut or blocked by a clot, the collateral channel can often provide sufficient blood to the area. Arterial anastomoses can occur around joints, where active movement may hinder blood flow through one channel. They are also common in the abdominal organs, the heart, and also the brain. And venous anastomoses are also common as well. So this will conclude the anatomy of the